I will now present to you a rough outline of the process development for groups who want to start manufacturing CAR T cells, as well as why that's challenging, and then talk about the solution that we have prepared to provide a shortcut through this process and generally make it easier to start offering these kinds of therapies. I will start by outlining the CAR T manufacturing development roadmap, starting from process development and all the way what needs to be done before clinical trials can be started, before then transitioning to introducing you to the cell manufacturing enabling package, and then presenting individual parts of this package in detail, and then also telling you on, if you are interested, how you can start with the CMEP, as well as commenting on what will remain to be done after you're done with the CMEP. To begin with, I think we can all agree that starting clinical manufacturing is not easy. And in particular, it's not easy to develop a manufacturing process and analytical methods. And it's in particular very difficult for clinicians who may be lacking the facilities, the experience in manufacturing, experience in QC, the regulatory know-how, as well as experience in how to properly train the manufacturing staff. And all of this together can make it a very daunting task to start offering CAR T cell therapies out of scratch. Anyone looking to start manufacturing CAR T cells will first need to focus on a suitable manufacturing process. And in particular, they will need to perform a feasibility assessment to see how feasible that process is. They will need to go through the process and define the basic steps of the protocol as so-called unit operations. And they will need to select a method to use for each of these unit operations. Further, they will need to go through the process engineering, which means that they will also have to find the optimal conditions for each of these unit operations. And in parallel, they will also need to develop analytical methods that will then later be used for quality control of the process. Further, they will also need to define a lot of things. For example, the target timelines. So when they want to achieve the different milestones in the process, they will need to define the desired clinical characteristics or quality target product profile of the cells that they are manufacturing, as well as the critical quality attributes or CQAs, which mean the criteria that they will use to establish how high the quality of the product is. They will need to define the context of use for their analytical methods, as well as choose the data that they want to report as part of analytics and formats that they want to report it in. They will also need to find the decision points and data collection points that will be used in the quality control, for release, for process control, for monitoring, and concomitant research. Further, they will need to lay out the plans for developing the manufacturing process, for developing the analytic process, then for transferring that technology to a manufacturing facility, as well as establish a clinical readiness plan. And of course, they will need to make sure that all of this that they are developing is aligned with the regulatory requirements. Of course, I've only given you a very simplified overview of these aspects that need to be developed and defined. And the process of development can take quite a long time, and we are still at the very first step that's required in this long journey. So once the manufacturing process and analytical methods have been developed, They need to be transferred from the process development or analytic development facility, which is usually referred to as the sending unit, to the future site of manufacturing, which is referred to as the receiving unit. And this also means that the PD and AD teams need to work with the MSAT team, which is the manufacturing science and technology team, who then transfer this process into a future GMP environment. This is, of course, not trivial. So they have to develop or establish something called a tech transfer plan, meaning that the documentation and records from the sending unit need to be transferred to the MSAT team. They need to perform a gap analysis, so to identify risks regarding facilities, regarding instruments, personnel, material, and so on. They need to develop a plan on how they want to transfer this, and they need to define the acceptance criteria, basically what it means that the tech transfer has been successfully performed. The MSAT team also needs to prepare a whole row of so-called GMP documents. They usually receive also support from the PD and AD staff in doing that. And of course, there are a lot of these documents and they usually have abbreviations, some of which you might be familiar with. So they need to write the protocol as so-called standard operating procedures, SOPs. And they also need to develop and write work instruction documents, bills of material, manufacturing batch records, and many, many other documents. And these draft documents are not yet GMP, but once they have been checked by the quality assurance and approved, they then become GMP documents. Following all of that, the know-how that has now been defined in these GMP documents needs to be also transferred to the personnel. So the future manufacturing and QC teams need to be trained. And this is usually performed at the receiving unit, meaning at the site where in the future they will actually be doing their work. And the acceptance criteria is usually a full process run, meaning that the trainees need to be able to demonstrate that they are capable of running the process that they have been trained. The final confirmation of a successful tech transfer is the so-called engineering run, which is basically a full-scale rehearsal of the manufacturing process. And that means that it's also performed at the receiving unit, but still not completely independently. So it's under the supervision of the sending unit, because the goal here is to confirm from all sides that the transfer has been successful, to test the general readiness of the people, but also the facilities, the instruments, the document drafts that are used to document the manufacturing, as well as to produce the first batches of the cell product that can then be used to test stability, both during storage, use, but also shipping, as well as to test the QC methods on. The process also needs to go through something called a characterization, which means that thorough understanding of the process is gained. And in particular, what's important to know is how the critical quality attributes of each unit operation affect the final product quality. In addition, it's important to do risk assessment and something called a failure mode and effect analysis, as well as defining the acceptance ranges for the product. Following successful tech transfer and achieving GMP compliance, the stability of the GMP product needs to be qualified. The analytical methods used for QC also need to be qualified, and it's actually more complicated if some novel methods are used which are not in the compendium of methods, because then also assays for qualification need to be developed. Further, process performance qualification runs need to be performed. And this means running the process using the finalized GMP documents under full quality oversight and demonstrating that the product can be produced at consistent and uniform quality over and over again. Additionally, something called the aseptic process simulation needs to be run. Some people also refer to this as the media fill run, and this essentially means using mock solutions, which are in reality media for bacterial growth, in order to demonstrate the sterility of the process. I hope that I've managed to give you a little bit of a feeling of why starting pharmaceutical level manufacturing might be a daunting task for clinicians. And however, many clinicians are still interested in providing cell therapies to their patients. And I will now introduce to you a shortcut that we developed to help people get through these development and also many of the achieving GMP compliance steps in order to be able to start as soon as possible qualifying and validating the process in order to be able to provide manufactured cells for a clinical trial. The Cell Manufacturing Enabling Package, or CMEP for short, consists of a cell manufacturing protocol as well as a protocol for flow QC, of on-site consultation of readiness to perform these protocols, in particular regarding the equipment that's described in them, a certified training, as well as support throughout this process and full documentation of all relevant activities, including the batch record templates that are used during the practical training, with the goal of bringing you to the stage where you are technologically ready to manufacture CAR T cells and where you can then begin this part of the journey into actually achieving GMP compliance. The protocol itself is a general protocol for manufacturing CAR T cells using the Miltenyi CliniMACS Prodigy Platform. It has 45 pages and has been developed through years of experience in our own cell manufacturing. And it has been established in clinical trials. It has also been designed for easy transfer and adaptation to local SOPs. So the format of the protocol makes it relatively easy to write into SOPs. Additionally, as well, we offer a protocol for flow QC using the MACSQuant instruments, also from Miltenyi. In order to determine the technological readiness of the site to actually receive the protocol, we have developed something called the pre-training checklist. And this is, as the name describes, a fairly long list. So this is just a small part, it's actually quite long, of items that for which we need to check whether they are present in order to determine whether the site is technologically ready to actually receive and run the protocol. And I think you have also seen this in the introductory part, that this is something that is quite a necessary part of preparing for tech transfer. And this is not very different here. Usually how that looks like is that we have field application specialists, and the local field application specialist will visit the site and will validate each of the points to make sure that all the equipment, all the reagents that are necessary are present in order to ensure the proper and smooth running of the training. The training proceeds in 3 steps. So it starts by a very introductory training, which we, of course, then intuitively named Instrument Introduction, followed by a theoretical background training, which we call the Essential Training, and then finally the Cell Manufacturing Enabling Training, which is the hands-on training intended to finally bring the staff up to the point and be able to actually manufacture the cells. The instrument introduction is very technology focused, so it's intended to transmit the knowledge of just the basic use of the instruments, just to make sure that the staff already knows this once they are receiving the higher stages of the training. And it's usually performed by the local Miltenyi specialist. And these are just introductions into how to operate the CliniMACS Prodigy and the MACSQuant Analyzer instruments, how to switch them on and off, basic maintenance, where different parts go on the instrument, and so on. So there is no deep knowledge, and there's definitely no protocol transfer at this stage. However, this is, of course, very necessary to save time and confusion and prevent errors at later stages. Following this, which, as said, takes place on site and is delivered by a local Miltenyi specialist, there is something called the essential training. This training is application focused, meaning that it brings a lot of background on the TCT process and on the CAR-T express mode for the flow QC. And this is a virtual training intended to transmit the knowledge, a broad knowledge and full knowledge of both the instruments, but also the standard applications. The training has 20 hours in total. But of course, I mean, no human can sit concentrated for 20 hours. So how that usually looks like is that we arrange with the host site different times when the different parts of the training will be transferred. And the goal there is, together with the instrument introduction, to have the staff ready both in terms of knowing the instrument, but also knowing the applications on the instrument and having a somewhat broader background knowledge on what's going on, so that they can then perform the final step of the training more readily and actually profit from it more. And this final step, which is really the focus of the cell manufacturing enabling package, is the cell manufacturing enabling training. And this is the actual technology transfer. So it's protocol focused, meaning that there's no theoretical background. There is no, let's say, variation. It's really training the people how to run the steps of the protocol, step by step. And the goal there is to bring them to the point where they can go through all of these steps independently and run them independently. So usually that means that they have something called a guided run, where the CMEP trainer is working with them and guiding them through, so supervising them through the process step by step. But then finally, they also need to be able to demonstrate that they can run something called the unguided run, meaning that they are capable of running the protocol without being prompted by anyone. And I like to kind of compare this a little bit on learning how to drive a car. So in the beginning, you of course just need to be able to know, how you switch off and on the car, where the trunk is, where the different parts are, the basic maintenance. But then you also have to have a background theoretical training on actually driving the car, on how to handle different intersections. But then the final part of the driving test is actually being able to drive the car and demonstrating that you can do it yourself. And if the examiner needs to help you, then you fail the test. And this is very similar how this three-step training is performed. And in particular, this final step, the cell manufacturing enabling training, means it's really a very, very practical training, and it's part of it is also a very, very practical demonstration by the trainees that they are indeed able to run this on their own. To come back to the analogy of car driving, this is also an important point of why the CMEP training is not your typical training. So if you imagine what a normal typical training, product training looks like that we offer and also other companies in the market offer, they are usually application focused. And they are intended to give broad expertise in use of the equipment. And the goal is to train on how to use the instrument and how to gain the full use of the instrument and how to be able to use all the different applications that are possible with this instrument. So the ideal audience for this are usually R&D or process development people. The CMAP, for its part, is protocol focused, not application focused. So the Prodigy has multiple applications beyond just manufacturing CAR T cells, but these are not addressed here. So the focus is really on that protocol, which is provided together with the training. And it's based on this protocol from the cell factory. And the goal, it goes beyond just the TCT process. It also includes the steps right before the protocol and right after the protocol. And the goal is really not to give expertise, but rather to make sure that the operators of the instrument know exactly how to follow the steps of the protocol. And of course, hence the ideal audience are not PD and R&D, but rather the actual operators who will then be running the instrument in a GMP facility. As said, after the training part of the training, there's also an exam, if you will, which is something called the post-training checklist, or rather the trainer goes through this checklist with the trainees. And they need to be able to demonstrate that they can execute these steps of the protocol. And the trainer is just standing beside them and noting what they are doing and validating whether they are able to perform each step on the list. With finally, once they demonstrate their capability of being able to independently perform the protocol, they are issued a certificate which contains the name of the employee, the name of the course, the version of the training, and the main topics that have been covered in both the theoretical and practical part of the training, as well as the date and site of the training, the date of issuing the certificate, and also the name of the Miltenyi employee issuing the certificate. And this is also very important for later steps because most authorities require proof of proper training of the staff. So these certificates can be used to verify when talking to the local authorities that the staff have been properly trained. Additionally, during the hands-on training, so during the cell manufacturing enabling training, the worksheets and batch record document templates are used because this is essentially a mock manufacturing process. And these documents are actually derived from the actual documentation used in our own CliniMACS cell factory, meaning that they are already in a format which is very friendly for adaptation to later GMP conform manufacturing and can be fairly easily used further once the GMP conform manufacturing starts to continue documenting the activities, meaning that there is significant relief in the burden of having to develop your own worksheets and your own batch record documents. And as said, so the final goal of all these activities and documentation is to bring the site really and the team to the point of technological readiness to start manufacturing CAR T cells, meaning that they know how to run the process, they know how to create batch records, and they can then start actually working towards reaching GMP compliance with then the ultimate goal of starting clinical trials for CAR T cells. Now, not everyone needs the complete cell manufacturing enabling package. And in particular, some users might have already had different access to analytical methods. And that's why we split the CMAP into two separate blocks. One for the CAR T cell manufacturing, which consists of a protocol and training, and a training kit, meaning that once the training is booked, the site also receives a kit containing all the reagents and chemicals that need to and consumables that will be required in the training. And separately from that, a flow assay a package can be booked, which then has the protocol and the training for the flow, QC flow part, as well as a separate training kit, which then contains all the consumables required for the flow training. Now, knowing what the CMAP is and what its intended purpose is, the question is, of course, if you are interested, how do you start this? And the first step is to reach out to your local Miltenyi sales representative. You can tell them what you're interested in and what you would like to do, and then they will talk to us, so to the training team, and we will then come into contact also with you. And before we can then start planning the training, we need to perform a site assessment. That means we need to be able to check whether from the technological side you are ready to actually receive this training. And then once we know that the site is actually suitable and that it makes sense to provide such a training, then we can discuss with you and our local sales representative in a kind of triangular communication channel and agree on the suitable timelines when you would like to host us and how we should logistically organize the training, whether you only need the cell manufacturing training or also the QC training, and when you can receive the different steps of the training and so on. Following that, we will send you a package, a physical package, which will consist of both the protocols, which you can then continue to use, as well as the kits containing all the reagents and materials that will be required in the training. Following that, there will be the 3 steps training. So you will probably have a visit from the local instrument specialist, just doing the introduction to the instrument. And then there will be these 20 hours of online learning, followed by a visit from a specialized trainer who will then actually run the cell manufacturing enabling training with the staff, followed by a competency assessment. And hopefully, once the staff have passed, we will again issue a certificate, and then you will receive certification for the staff that they have been properly trained to run the process, and that they are also capable of doing the process themselves. The ultimate goal is to bring you to the point where you don't really need to do any process development and where you have successfully transferred the protocol to your site. The protocol is based on SOPs from our side, so it's fairly easy to translate into SOPs. And as I already said, during the training, there will also be batch record templates, document templates that will be used, which means that it should also assist with these initial parts of achieving GMP compliance, in particular, give you templates and formats which will make it relatively easy to prepare GMP documents. It will also give the staff the know-how in how to perform the actual run. This means that after that, you will be able to start doing the engineering runs and also to start characterizing the process. Now, of course, this is just the beginning of the odyssey towards CAR T cell manufacturing, because after all of that, you will need to validate the process and to get clearance from the regulatory authorities to actually start offering clinical trials. So the CMAP will really bring you through this period of protocol development, of process development, sorry, and also through a big chunk of successful, hopefully, technology transfer. But then the validation needs to be done at the site where the manufacturing will actually be run, and it also needs to be done by the organization which will be running this site. So this is something where we cannot which we cannot do for you. But one of the features of the CMAP that I also mentioned is open communication channels and constant support, meaning that we will, of course, stay in touch with you also after you're done with the CMAP training, and we will support you throughout. So the goal there is really to send you on your journey, but not to send you alone. And with that, I would like to thank you very much for your attention and for taking your time.